So hello everyone and welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Dan Ma. Professor Ma received her BSc from Zhejiang University and her PhD from Case Western Reserve University. She is currently an assistant professor at the Department of Biomedical Engineering and the School of Medicine at Case Western Reserve University. She has received the Hiroki Fujita PhD Award at 2012, as well as the ISMRM Rabbi Young Investigator Award at 2016, and also the Outstanding Teacher Award at 2018. She is an ISMRM Junior Fellow and serves as on the editorial board of Magnetic Resonance in Medicine. I would just like to remind the audience to please address any question you have using the Q&A box. Professor Ma, thank you very much for coming here today. The virtual stage is all yours.
Thank you all for your introduction, and thank you for inviting me to give this talk. Today I'm going to talk about an MR imaging technology called MR fingerprinting. I'm going to introduce our effort of translating this basic technology development, imaging technology to more of a clinical setting. MRI is an imaging technology that widely used in clinical settings, basically because MR can provide very great soft tissue contrast. A major limitation of the current MR scan is that most of the MR scans currently used in clinical settings only provide qualitative measurements of limited tissue properties, such as T1 weighted or T2 weighted images. So here's one example of T2 weighted image. So we call it a T2 weighted images because this image shows you the relative contrast between the lesion and the surrounding tissue types. So in order to describe this lesion, we typically call the lesion is hyper-intense or hypo-intense as compared to the surrounding tissue types. Well, this is a basic description of relative contrast, but this contrast may vary from different scanner settings or different days, even scanning with the same subject. Because this contrast is relative contrast, and we are not be able to get any numbers from this image, the diagnosis currently still heavily relies on subjective evaluation by radiologists. So also cause a large challenge of making this MR scan standardized because it's really hard to control the contrast across different vendors and different sites. Another challenge is that it has been reported that this large variation from the weighted images may also cause instability in the downstream image analysis, which means that image features we got from the post-processing are highly dependent on the imaging settings and the contrast variability from those images, and altogether may cause the uncertainty for the clinical support. So this is the problem, especially for nowadays. We start to use this digital images, big data analysis, and the precision medicine for the clinical decision support. If we want to improve the overall consistency or certainty, I think the best way is to improve from the very beginning of this pipeline, which is the weighted images. So we're trying to improve how we could generate images, and following that, we might be able to improve the stability of the image analysis and also certainty of the decision support. So how do we do that? So let's go back to this image. Instead of generating a series of T2 weighted images, alternative way is to use a quantitative scan to quantify T2 number from this image, from this brain. So for example, we can quantify T2 at different tissue types and put a number in, for example, healthy tissues, healthy gray matter, healthy white matter, and also the number, T2 number for lesions. By using this number, so these are all quantitative numbers, which are more intrinsic, more of intrinsic tissue properties. This number are less dependent on system variations or acquisition settings. If we are able to use this number for lesion detection and characterization, the decision support may be less dependent on subjective diagnosis as well. So, and also because we are using MRI, MR is actually a very versatile tool that is sensitive to lots of tissue properties in addition to T1 and T2. For example, flow, perfusion, diffusion, susceptibility, all these parameters demonstrate different aspects of the tissue. So if we are able to quantify those parameters, we could potentially improve the specificity and sensitivity to different disease status. So this is the background, and the goal of developing MR fingerprinting technology is to provide a clinically feasible quantitative MR scan. So here, more specifically, we're trying to develop a fast, robust, and multi-parametric quantitative MR scan. So fast, meaning that we hope this scan can be applied to clinical settings and can be implemented to the currently very busy clinical workflow. Robust, meaning that we hope this scan can be applied across vendors at different sites, but providing consistent results, and can also be applied to image different parts of the body. Multi-parametric is a way to improve the scan efficiency. So if we're able to quantify multiple tissue properties from a single scan, we don't need to worry about image co-registration or motion during the scan, such type of confounding factors in the downstream. So this is our general goal of developing MR fingerprinting. So the name or the concept of MR fingerprinting is analogous to the conventional fingerprinting technology. So basically, if we have a fingerprint from a person, by using pretty advanced template matching or matching technology, if this sample can be matched to one fingerprint from a comprehensive database, a host of additional information from this person, such as name, phone number, and address, can be extracted simultaneously. So MR fingerprinting followed very similar structure or idea. So it started with the signal acquisition. We don't have any requirement or constraints for what shape of the signal should be, as long as the signal from different tissue types are different and incoherent. So similar to the conventional fingerprinting technology, we build a database, or we call it dictionary, that this dictionary contains signals from different tissue properties. We could build this dictionary using block simulation or other types of model. And then the next step is to match the signal to the dictionary in order to find the match. And once the match is identified, the underlying tissue properties such as T1, T2, or perfusion, as well as some system confounding factors, for example, B0 and B1 field, can be quantified simultaneously. So this is the general diagram of the MR fingerprinting framework. So how do we implement that? This is another very simple example. We acquire a series of images, and from each pixel of this image, we can get the signal evolution along the time. The signal can be arbitrary, the shape can be arbitrary. But if we build a dictionary and match the signal from to one elements or multiple elements of the dictionary, we will be able to quantify multiple tissue properties at the same time. So we've been working with industry to develop a clinical feasible MR fingerprinting scan. So here is one example of 2D MRF. In-plane resolution is 1 by 1 millimeter, scan time 20 seconds. So after this 20 seconds of scan, T1, T2 maps can be generated online, simultaneously. So here are 3 examples of different of T1, T2 maps from different views. Following that, we also developed 3D MRF protocol, which is able to quantify T1, T2 problem density maps simultaneously from a single scan. So these are all the maps from different views. And from the same data sets, gray matter, white matter, CSF segmentation can be performed. So all these maps, because all these maps are from the same scan, there's no registration issue at all. All the images are inherently co-registered altogether. The examples I provided are mostly T1, T2 maps. As I mentioned, MRI can quantify lots of other tissue properties as well. So the benefit of the MR fingerprinting framework is that you can basically design, you can basically have your own MR fingerprinting recipe to quantify other tissue properties. So here are a few examples that other groups have developed multi-parametric mapping based on MR fingerprinting. So on the left, this shows the MR vasculature fingerprinting, which is able to quantify cerebral blood volume, vessel radius, and oxygenation level all at the same time. This is more of a recent study on fingerprinting-based assessed imaging, which also able to quantify multiple tissue properties, assessed-based APT-related tissue properties from a single scan. This study also compares the conventional MR fingerprinting way, which includes block simulation and template matching, with more of a recent deep neural network quantification method. So both methods should work, but it's really amazing to see the comparison of the conventional way of doing mapping versus the deep learning way of doing mapping. As also mentioned, in the dictionary simulation, we try to simulate signals under the certain acquisition parameter settings, tissue parameters, and also system conditions. So that includes B0 and B1 inhomogeneities. So if we simulate the signal by considering those confounding factors, in the mapping step, the MRF will be able to quantify tissue property maps as well as the system confounding factors altogether. The benefit of doing that is that the resulting quantitative tissue maps will be less affected by those confounding factors. So one way of applying this concept is that there's a group of applying this MR fingerprinting to ultra high field. So as you can see from the maps, T1, T2 maps were quantified along with the B1 map. So as a result, the T1, T2 maps are less affected by this very severe B1 inhomogeneity. Another benefit of mapping with confounding factor is that the resulting T1, T2 can be more robust as well. So for example, if the MRF scan is implemented across different vendors or different sites, by considering those field variations, we might be able to get more robust or reproducible quantitative mapping. So apart from neurals, there are also other studies applying MR fingerprinting to other part of the body. For example, cardiac imaging, liver, these are hip imaging, and the breast imaging. I want to emphasize that all these are quantitative maps, meaning that the numbers from all these images now have meanings. these numbers are all more intrinsic to tissue properties, which is supposed to be more reproducible and less affected by confounding factors or acquisition settings. So this is this is a review recently published in JMR. Pretty great summarize the technology that have been developed using MRF based concept, which include ASL, B1 plus mapping, cardiac access, and MT. As I mentioned, based on this very flexible concept, the different variety of the MRF based sequence can be developed, and different types of tissue properties, apart from T1, T2, as I mentioned, perfusion, diffusion, have all been studied in this current setting. So now that I introduce this quantitative framework, which is able to quantify multiple tissue properties in a short scan time, and we start to see more and more tissue property maps nowadays. And all these maps were quantified from the same scan, and more efficient. And the scan time is very efficient. Now what? What's next step? I've been asked many times, what's the next step, or what's next of the MR fingerprinting? I really enjoy reading this review and a commentary article from Radiology Journal. And the title is Reproducing Fingerprints, a Step Towards Clinical Adoption. And first of all, this article kind of pointed out the promising and the significance of this technology. We are at the pivotal point in the clinical setting, in the clinical use of MRI. As never before has quantitative measurements been so readily available. MR fingerprinting may be the technique that makes common use of quantification a clinical reality. And this review, of course, did not stop at here. It also brought up a very important question. There has been pretty slow translation of MRF to clinical applications since 2013, when the first paper was published. So what's the key challenge of translating this technology to clinical settings then? In summary, there are three critical points or questions. First of all, MR fingerprinting scan involves scan design, dictionary simulation, and template matching, or are more complicated than the current MR scans that have been implemented in this in the scanner. It can be the scan time can be long if we are going to scan a large tissue. If we need a large tissue coverage, of course, the MRF also requires relatively higher computational power in order to again, create the dictionary and generate maps. And also, it's also challenging if we really want to make sure that everything, all the maps are generated real time after the scan, so that this protocol can be incorporated into the more of a busy clinical workflow. The second point is that because MR fingerprinting is a quantitative scan, we have to understand the accuracy, precision, and generalizability before we apply a quantitative scan to clinical settings, because now the numbers are matters. And finally, and this very important question is that what's the added value? This may not be the only question for MRF, but in general, for all the quantitative scans, given that the current MR is already pretty successful as a qualitative measurements, so what's the added value from the quantitative maps? This is an important and very challenging question to answer. So in terms of the technology, there has been a large effort of sequence optimization, for example, to try to reduce scan time and optimize the scan sensitivity to various of tissue properties. In terms of the fast mapping or computational speed, there are also studies on data compression, fast mapping, and of course, machine learning to really speed up the mapping right after the acquisition. So this is one example of the deep learning MRF, which directly predict the maps, T1, T2 maps, from the reconstructed images. So this is work. This is the work developed by Dr. Chen. By using the deep learning MRF, they were able to generate T1, T2 map of the entire brain with 1 millimeter isotropic image resolution in only 7 minutes. In terms of the sequence optimization, MRF sequence optimization is actually a very challenging question. Here are a few main points. So first of all, because MRF, because there are variable flip angles, TRs, or all these different type of acquisition settings, there could be thousands of parameters to be optimized. In the meantime, during the acquisition, or especially in vivo scan, the current MRF implementation applies very high undersampling factor, which means that we are dealing with aliasing artifacts in all the images. And the aliasing artifacts is not cannot be fully represented by white Gaussian noise model. And finally, the matching relies on a template matching, which is also a nonlinear process. So based on the conventional sequence optimization framework, it is really hard to optimize MR fingerprinting scan. So this is more of a recent framework developed in our lab. We rely on mathematical model to synthesize all the images from a brain phantom. But during the simulation, we simulate the errors from actual undersampling trajectory and the B0 field. The main purpose of doing that is to kind of predict what's the actual error from the actual in vivo scan. After we generate this prediction of T1, T2 map, we use this error, which is more of a direct estimation of the results in the cost function. We build this error in the cost function and use the total scan time as a constraint. In collaboration with Microsoft quantum computing team, we've also developed this optimized optimization algorithm to optimize the MR fingerprinting scan. So the entire framework incorporated physics-based modeling and also the very powerful optimization algorithm developed by the Microsoft team. Why do we think this physics-based model are important? So here is one simulation results demonstrating the T1 and T2 maps from different settings. So here are T1, T2 maps from a model by considering only undersampling artifacts. You can see that there are some artifacts, residual artifacts from T1, T2 maps. Here are the T1, T2 maps derived from the model by incorporating undersampling and B0 inhomogeneities. These two scans are from the same with the same scan time, but clearly there's more artifacts shown on the more of a comprehensive model. So here we see the aliasing artifacts from undersampling and also this shading artifacts that is not shown from the undersampling only model. So here is an actual in vivo scan that we got by using a truncated MR fingerprinting scan. So these two have the same acquisition setting. So here we can see that by using this physics-based model, we are able to predict those aliasing artifacts and the shading artifacts from the actual scan. If we use those estimations, so here I want to point it out that using this model, we are able to direct predict error from the in vivo scan. This is already a good quality analysis or uncertainty analysis of the established sequence. Well, we can also incorporate this error analysis into the optimization framework and directly minimize the quantitative error. Here are the results from the optimized scan. So here, B0 variations were incorporated. So these 4 maps were actually from totally different B0 directions and variations. Well, we can see that optimized scan, optimized sequence is able to get rid of those shading artifacts here, as well as the aliasing artifacts. So here is the corresponding in vivo result we got. Again, there's no shading artifacts at all from the optimized scan. So we hope that this physics-based model, physics-based model-based optimization will be able to really improve the robustness of the MRF for the actual in vivo scan. Okay, so next point is accuracy, precision, and generalizability. This is very critical for quantitative MR scans, and it really takes time to validate everything. So we've been using this ISMRN NIST phantom to estimate accuracy and the repeatability of MRF scan. As you can see, this phantom has different layers with wide range of T1, T2. So this is the T1, T2 relaxation phantom. Then I believe NIST also provide other the phantoms for other tissue properties as well. So this phantom was used to evaluate the repeatability in 5 different sites using 2 different scanner models, and we can see the pretty good repeatability across these sites. And these are basically phantom reproducibility scan. Recently, there are also multiple publications about reproducibility and repeatability of MR fingerprinting in the human brain. This is one example. This paper published in Radiology. 10 healthy subjects were scanned in 10 scanners with different models and in 4 different sites. So this study was performed in a single vendor. From this paper, we can see that the repeatability and the reproducibility of MRF is quite substantial, even in the in vivo scans. So 2 to 3% for T1 and about 8% for T2 for repeatability. Similarly, 3.4% for T1 and 8% for T2 for reproducibility. Another study published in Neuroimaging, again, also repeatability and reproducibility, but I want to emphasize that this study was performed in a different vendor. So MRF has been studied, the repeatability and the reproducibility of MRF have been studied in 2 different vendors. So again, in vivo human brain, 5 different scanners. So here they study the repeatability and the reproducibility in 2 different fields, 3T and 1.5T, with different models and 2 different sites. Again, the similar repeatability and reproducibility levels were reported in a different vendor. So in the last section, I want to talk about what's the added value. This is actually, again, this is a very challenging question to answer, and it actually takes a long time to really validate and prove that. So here I only list the potential clinical questions or benefit of using quantitative scans as compared to qualitative scans. In terms of the generalizability, if we prove that the repeatability and the reproducibility of such quantitative scan are pretty good, the scans can be used across vendors, hopefully, and multi-site study. for the robustness, this quantitative scan can be applied in ultra high field and ultra low fields. For the precision and reproducibility, if the precision of the in terms of the technical variation is well controlled, this quantitative scan may be used for longitudinal study to monitor the treatment response and predict the outcome. This high precision and reproducibility can also be used for measurements and data standardization. So hopefully, once this quantitative MRF scan is implemented in different vendors, we'll be able to get more of a standardized data and build a bigger database for quantitative analysis. And finally, it's more of a question whether the quantitative MR can provide higher sensitivity and specificity as compared to qualitative MR imaging scans. If we have quantitative numbers, are we able to identify global changes easier than qualitative scans? As I mentioned, qualitative measurements heavily relies on relative contrast or relative comparisons. If we have absolute numbers, are we able to identify global changes? Whether the quantitative numbers are more sensitive to identify subtle lesions or maybe identify abnormalities in normal appearing tissues. And finally, if we have all the numbers for different type of diseases or lesions, are we able to use those numbers to characterize lesions and better classify different maybe disease types? So because I put a question mark here, it's because that really requires a lot of clinical studies and larger patient numbers. So here I only want to show you some preliminary results to identify maybe promising results, promising directions of quantitative scan in disease detection and characterization. So I've been working with Cleveland Clinic Epilepsy Center of applying 3D MR fingerprinting for epilepsy patient scan. So this is one example of epilepsy patients. So this patient has heterotopia around the ventricles, which we can clearly see already in the T1 weighted images. But there's no way to identify at what part of this lesion may cause seizure. So from the T1 map, we start to identify this substantially increased T1 in certain area in one corner of the heterotopia. So again, this finding is not shown in the conventional T1 weighted images. So this patient later had an invasive SEEG, where the EEG was implanted into different parts of the deep brain, and in order to identify the actual seizure onset location. So while this red dot showing the seizure onset location, which actually matched the T1 increase area here. This is more of a reference or validation of our MR fingerprinting finding, while this one is more invasive. If we're able to identify the abnormality from more of a non-invasive imaging technology, that would be great. This patient later did a surgery to remove this part of the lesion and has become seizure-free. Because it's a quantitative scan, we could also characterize lesions in the epileptic nodule and non-epileptic nodule and the surrounding normal tissues. So here includes white matter and gray matter. And using the T1, T2 numbers, we'll be also able to characterize the lesion. So as you can see, epilepsy nodules here has a very distinct cluster as compared to non-epileptic nodules and normal tissue types. This is another example of the epilepsy patient who has right temporal epilepsy. This patient has enlarged amygdala. While in the T1 map of this patient, we start to see this pretty subtle tail extending from gray matter to white matter. This also a new finding that is not identifiable from the weighted images. This tail is further highlighted in the gray matter fraction map, where because we in the fraction map we remove the white matter sections, we kind of highlighting the contrast between the gray matter fraction and white matter. So here is the highlight. This is also a new finding. After removing this part of the lesion or tissue, this patients also become seizure-free. So this is more of an individual level finding. We of course need more patients in order to further validate the finding and have more of a statistical numbers to characterize the lesion in the future. So another thing that I want to point out is that now we have quantitative maps. In addition, so as apart from generating more quantitative maps, we also want to bridge the gap between quantitative maps and the clinical decision making. So here, so we in collaboration with university hospitals and Cleveland Clinic at Cleveland, we also developed this more of an imaging and post-processing pipeline to bridge this gap. So in the pipeline, the input can be clinical MR and quantitative maps, and then followed by pre-processing, integrated analysis, and feature extractions. After the feature extractions, a lot of other feature analysis can be performed in order to support the decision making. One example, apologize, this very busy slides. This is the our initial implementation of this imaging and post-processing pipeline on brain tumor. So though this slide is very busy, the basically comprised of three basic parts. One is the pre-processing, so-called pre-processing is the imaging part, which generates multiple quantitative images. And then the second one is the conventional post-processing, but based on the quantitative maps, which are supposed to be more stable and reproducible. And based on that, we will be able to get higher order features from those images, and use those features. We could evaluate different, let's say, different tumor types, and correlate the features to outcomes, for example, recurrence and survival rate. Here's one initial pilot study of using MRF plus radiomics of classifying different tumor types. My collaborator also correlate this type of image features to survival rate of GBM. So here they were able to find some pretty good differentiation between the poor survival and better survival. Again, these are pilot study using limited number of patients. Of course, using this preliminary data, will hopefully we can include more, recruit more patients and have more of a statistical analysis. So by bridging the gap between imaging and the clinical decision making, another important part is that there's also possibility of incorporating multimodal, multimodality imaging technologies. So this is the study from University Hospital Essen by incorporating MR and PET MR together, and use the machine learning to incorporate all the features and predict mutation status based on different genetic settings. So again, this is also a promising direction that bridging the imaging technology or imaging information to more of a clinically friendly decision supporting tool. Again, this is a summary of what I see opportunities for quantitative MR. We have a pretty flexible framework to generate more repeatable and hopefully more specific information from quantitative maps. This is not just limited to T1, T2. There are also other functional and physiological related tissue properties that we are able to quantify. All this information can be connected naturally to the image analysis pipeline, which is a downstream analysis, and where we could use a statistical model, machine learning, or even multimodal and multi-scale data to have more of a multi-dimensional information. And hopefully we will be able to use all this for the decision support. I gave some examples of lesion detection and the disease characterization. Of course, there are also following prediction of outcome and treatment planning. So again, I believe that there are also lots of opportunities in terms of the entire pipeline. Okay, that's all I have. Thank you.
Great. Thank you very much, Dr. Ma, for this great talk. We now move to the Q&A phase. So anyone can just use the Q&A box to type your questions. And on the meanwhile, I'll just quickly ask a technical question. How big are your dictionaries? How many entries you have, or how many elements?
Well, this is a good question. It depends on how many tissue types you want to quantify. So the dictionary typically is a 2D or n-dimensional matrix. One dimension is number of images. So for example, this MRF scan, number of images can range from hundreds to thousands. This is one factor of the dictionary size. Another factor, as I mentioned, is number of tissue parameters. So for example, if I only want, only plan to quantify T1 and T2, there are 2 dimensions. But when it goes to more of a more complicated model, including multi-compartment model, CEST model, the number of tissue parameters can be up to 7 or 8. So at some point, dictionary simulation might not be a feasible way. That's why lots of research propose more of a compressed dictionary simulation or directly machine learning or deep learning type of prediction that kind of avoid huge amount of computing requirement for dictionary. Another factor that determines the dictionary is the step size. So how fine or how accurate or how precise you want your result is. For example, T1, if the step size is 100 millisecond, so that means how many what's the resolution of your tissue. That heavily also affect the size of the dictionary.
Thank you very much. And so for a typical neurological application, if you only quantify T1 and T2, we're talking about tens of thousands, millions, out of curiosity.
Yeah, I think currently, if you want to keep about maybe 5% of step size, that can be hundreds megabytes, yeah, hundreds of thousands elements. There are also other ways for dictionary compression, for example, the SVD compression or other things. So just to make sure that the dictionary size is not too big, and also because the next step is template matching, that also the size of the dictionary also affect the mapping speed.
Great, thank you. So we have quite a few questions piling up. First question from Alex. Alex, can you please unmute?
Yeah, thank you very much for this excellent presentation. My question is very simple. In your opinion, what is the advantages and disadvantages of fingerprinting compared to synthetic MRI? Because I think that both techniques are available on different vendors, but it's not clear from a clinical point of view which is the advantages of one to the other.
Yeah, I think the synthetic MR is quite a good technology as well. It provides multiple contrasts from a single scan. The main difference is that MR fingerprinting is a quantitative scan that generates quantitative maps, so for T1, T2 maps, while synthetic is trying to produce weighted images that maybe closer to the current MR standard. Yeah, that's the main difference. I think both have advantages. I think for the synthetic MR, you will be able to get all the contrasts you want. And I think the disadvantage is that images are still weighted images, so you're still looking at contrast. So may not be like substantially different from current standard diagnosis, or maybe there's still some challenge of standardizing those images. That's just my point of view.
Yeah, thank you. But synthetic MRI also can provide T1 and T2 maps with a different, I think, way of obtaining this information. But I don't know if any study had compared the T1 and T2 maps obtained with these two different sequences. I'm not aware of that.
Not aware of that, yes. So as you mentioned, yes, the synthetic MR typically first to quantify T1 and T2 maps first, and then synthesize different contrasts. Both MRF and other type of method can do that. I'm not aware of any studies comparing the underlying T1, T2 from synthetic MR technology to more of a quantitative measurements, though.
Thank you. And then the next question comes from Beth. Can you please unmute?
Yes, I can. Thank you. Thanks for that really nice talk. I was wondering if you can tell us a little bit about whether you're able to look at MRF pathology correlations, and in particular with regard to like being able to use the MRF generated maps to differentiate different aspects of underlying pathology in multiple sclerosis lesions.
So we haven't not done any correlation for MS patients. We started to do some correlation analysis for in the brain tumor setting, not at the level of pathology though. We're trying to correlate the MR fingerprinting features to say a diagnosis outcome, different tumor types, recurrence, survival rate, and the different genetic mutation levels. I think this is the pathological correlation is a pretty important direction as well. It might need more of a final resolution or more specific features. Yeah.
Yeah, I know. I mean, I asked this very specific question, I guess, because I was working with the team, with Mark Griswold and the group years ago, back when I was at the Cleveland Clinic, and we had initiated this type or this particular project, and I just wondered if it kind of got off the ground or not. So it sounds like perhaps it didn't, but I do think, what do you, in terms of the promise for the technique for being able to do something like that, it sounds like it would be applicable perhaps to that type of, like that might be another one of the added values of this approach, is that we can get more pathologically specific information in a reasonable time frame using this type of technique.
Yeah, definitely. As far as I know, maybe not in the neuro, but Dr. Vikas Gunali has been collaborating with Dr. Anand Madhabushi from case, trying to correlate the prostate MR fingerprinting images to more of a digital pathology. So they recently published a paper on that. Yeah, that's definitely a good direction.
Excellent, thank you.
And next question from Ross. If you could please unmute.
Okay. Yes, thank you for your talk. I noticed in the large table of applications you had in about the middle of the talk, you mentioned, well, there was the entry about motion. I was wondering if you can say something briefly about what impacts motion might have on this technique, or if it's lack of impacts, especially in comparison to the others. Obviously, it's a single scan. You're not requiring separate B1 maps and things like that. So that's going to be a plus, but I just wondered how motion impacts the overall estimation of these quantitative parameters.
So the subject motion, if we're talking about motion, not periodic physiological motion. So the subject motion, there are a few studies kind of evaluate what's the effect to the quantitative maps. So this motion, because we use the spiral acquisition, and all the maps are from a long series of images, and the impact is more complicated as compared to motion artifacts that may affect the T1 and T2 maps, the values. So a few groups have been working on to develop more of an uncertainty evaluation on how to track the motion effect in the MRF scan. So this is still ongoing research, I believe. In my very original study, we also study what's the, so because of this template matching and the dictionary simulation, MRF is more robust to short-term motion. For example, if the motion happened in the middle or at the end of the scan, 20%, in the resulting T1, T2, we don't see much of the motion artifacts. So it's more robust, but there are also some potential change in T1, T2 values that we have to pay attention. In terms of other physiological motion, for example, cardiac motion, and yeah, so there are lots of studies on how to use more of a gated or ungated way of correcting those motion or track those motion.
Okay, great. Thank you.
Thank you. Next question from Larry. If you could. Larry, can you please unmute?
Sorry about that. Yeah, I was just curious. Thanks, Dan, for a great talk, as always. I was just curious why you picked the epilepsy of cortical dysplasias and heterotopias as your sort of first clinical trial with this. It would seem that that particular application puts a high premium on spatial resolution and essentially contrast to noise. And I'm just wondering if you pay a penalty in those to get the maps.
So yeah, so in terms of first the clinical target, our first clinical target is we've been working on brain tumor and epilepsy. So it's not just the epilepsy, but epilepsy project. At the moment, I developed this isotropic whole brain MRF. So we hope to see more of a whole brain clinical applications. And the epilepsy question is also pretty important because the epilepsy lesion can be very subtle. As you mentioned, it has high requirement of the resolution, but also specificity. Because some of the epilepsy lesions are mainly in the gray matter, white matter boundary, only cause the blurriness, and it's really hard to tell. Also, as I mentioned in the example that although are able to see the lesion in MR, but they're not able to tell apart or they need more of a specific way of identifying the epileptic nodules. So I think this is first of all, it's very interesting topics, and it's very, it has clinical significance because once we identify those focal lesions, those patients can become seizure-free after the surgery. So yeah, of course, this is a very challenging, clinical applications. We are still trying to improve the resolution. So to really looking at the cortical dysplasia. So currently 1 millimeter isotropic, still not that sufficient to identify the especially cortical layers. So there are still a lot of work to do.
So would you say then you're sort of more focused on setting it up for a computer-aided diagnosis to sort of take over and find these subtle lesions? I mean, that would certainly make sense.
Yeah, so this is also, I got this knowledge from my collaborators as well. So because this lesion is pretty subtle, it's one thing is challenging to just simply looking at maps to identifying the subtle lesion. So they already have the epilepsy field already has more of a specific image post-processing tool to identify this. So if you are able to get more, if we are able to use quantitative maps, I think is really beneficial of combining this quantitative images and as well as more of an automatic computing tool to identify subtle lesions.
Cool, thanks.
So I guess, yeah, the key point is that I think providing more and more maps may not be the end of the story. I sat with the radiologist and so the thing that I feel is that our goal is not providing them more and more maps. Yeah, so definitely the downstream is also an important part.
Thank you. Next question from Bruce.
Hi there. Fantastic talk. Really enjoyed it. Just to kind of jump on Larry's point, it seems like applications, say in epilepsy, but outside of that, more broadly, where you've got multifocal lesions, maybe patients with NF or the like, where we have reason to believe that, a small subset of them have these, unique, bad characteristics. Maybe they have malignant potential. Maybe they're generating epileptic foci. Maybe a domain where the quantitative numbers, really may be able to distinguish amongst lesions that otherwise kind of look roughly the same, as opposed to say, your Taylor migration abnormality case, where really the numbers you get basically is just the mix between gray and white matter, and maybe there something like a high resolution image might, kind of tell the tale. Just as an aside, but I was actually thinking of a slightly different, question. I had a birthday recently, so it's inspired me to ask an old man question. So, people have been measuring T1s, back since Damadian, so that's 50 years ago. And of course, the technology for measuring T1 has evolved, over, and T2 has evolved over the years. But, all of those studies going back, 50 years have shown that, there's a high degree of, kind of tissue heterogeneity, as well as noise, if you will, based on the measurements. So, kind of what makes you most excited in your data or in the methodology that makes you think that, today, we've really got a chance to kind of weave these techniques into clinical practice in a way that we've kind of had at least some capabilities to do, and have always been fighting against the biological variability as well as the technical variability in the past. Why is this going to be, different than it was, back when Ray Damadian said we were going to be diagnosing cancer based on measuring T1?
Yeah, very good question. I think
An obnoxious question, but I was curious to see how you handled it.
I think T1T2 maps may not be the end story as well. So I think the benefit from MR fingerprinting is that we kind of, so first of all, we are able to consider all the confounding factors along with the tissue properties. So in this way, maybe not just the B0 and B1, but also slice profile and other more complicated confounding factors. So in that way, it's possible to have more of a repeatable or reproducible tissue mapping by considering all these confounding factors. Another thing that I'm thinking is that MR fingerprinting is kind of a way to encode multidimensional data and quantify multidimensional data at the same time. So instead of quantify one thing and assuming other factors are not significant, we kind of consider all the things altogether. It's more of a multidimensional analysis or encoding. So I think this way you will be able to get better estimation of the contribution, because as we know, the MR signals is always affected by a lot of tissue properties. It's really hard to quantify only one by assuming other things are not affected. And finally, I think in terms of the brain tumor and other disease analysis, I want to emphasize that T1, T2 are the example that I made. Of course, there are also other more specific tissue properties that we may be able to quantify. So I think the way to see that is, for example, let's say one tissue properties, they of course have lots of variations in terms of the physiological variation and other variations. The way we quantify this and trying to remove other confounding factors, then the result can be more specific. For example, if we say T1 increase, or let's say other tissue property increase, that can be from this, can be from that. But if we kind of quantify other properties together, we may be able to get more of a specific estimation or explanation, other than having more confounding factors along the way. So speaking of physiological variations, we actually start to build this database of the healthy subjects, T1, T2. And we start to see this age-based, physiological-based, or even cortical area-based variation for T1, T2. I'm sure there are previous studies on this cortical or functional region-based T1, T2 variations. So I think building, if we are able to get this more of a reproducible scan that can be applied for multi-center, we might be able to get a baseline, normative baseline of quantitative values across different age or genders and other stuff that may be able to also remove those physiological variations from the disease uncertainty or diagnosis uncertainty.
Great. Thank you very much. Good answer.
Okay, next question from Eve. Can you please unmute?
Hello, can you hear me?
Yes.
So I was just wondering about the potential applicability of using this for something like repeated mild traumatic brain injuries or concussions. I know that may be a challenge, but we know that typically scans do not do well for mild traumatic brain injuries or concussions. But it's always good to know if there's a possibility on the horizon.
Mild traumatic disease. Can you repeat your question? Sorry.
I'm wondering about the potential applicability of using MR fingerprinting for identifying possible evidence that would suggest a history of mild traumatic brain injuries, like repetitive traumatic brain injuries. That's a population with which I work, people who sustain many brain injuries that are often mild in nature, but clearly cause some type of damage to the brain that has long-lasting effects. So just wondering about if this is something that you had ever thought about or maybe a possibility.
Well, first, I've never really thought about this, but second, I think that might be related to the point that I brought up in the potential advantages of quantitative MR, is that especially for this long-term change of the brain, it might be hard to evaluate in using more of a qualitative contrast measurements. But if we're able to, so first of all, if we have the baseline database, like more of a healthy brain, we might be able to identify more of a global change or subtle change in the normal appearing tissues. Is that yeah
I mean, yeah, I think that could be, that's what I'm wondering, if something like that is possible. I mean, what we know about concussions or mild traumatic brain injuries, I can actually, so, you know, talking to a black box. What we know about concussions or mild traumatic brain injuries is really that if you take someone who you know has sustained one concussion or repetitive concussions for that matter, and you just give them a regular scan, you may not see anything. And we realize that's partially a limitation of the technology that we have. And I'm just wondering if this is something that potentially would be amenable to determining more potentially diffuse effects that may be long-term.
Yeah, I actually think that diffuse disease can be, so quantitative measurement can be a good tool for diffuse disease and maybe a measure of global changes. Although I haven't really done any study about this.
Okay, well, it'd be great if you did, let me know. Thank you so much.
Right, last question from Brian.
Hello?
Can hear you.
Can you guys? Okay, great. Hey, Dan, very nice talk. Just very tiny technical question. I didn't see any lipid maps in your images. Are there plans to tackle that little nitty-gritty issue?
Well, I remember there is a publication about this. I'm not sure, but I actually heard of this. I personally haven't done any lipid simulation yet, but I'm sure that there's possibility of incorporating this component into the scan or into the simulation.
Cool, that's great. I saw you had the B0 inhomogeneities, and it's kind of the same part of the model. So cool. Thanks.
Great. So thanks again, Dr. Ma, for this great talk, and we hope to see you soon in person.
Thank you.
Maybe.
Thank you
Very much. Bye, everyone.
Bye.